Good morning. My name is Carrie, and together with my colleague Nima, we'll be spending the next couple of hours describing and familiarizing you with the IEDB 3.0 search interface. So I'll start with a brief overview of our part of the day. It'll be broken up into essentially three parts, and these will be punctuated by breaks and group lunch and that sort of thing. But basically, the first part that we'll be going through this morning will be an overview of the search interface, looking at the results page, familiarizing you with all that. Bjorn has done a wonderful job of starting to introduce those images to you, so you've already seen them. We'll talk about some really important functional aspects of the database, tree views and finders. Again, they were introduced to you in Bjorn's talk, so I'll go into them in great detail so you understand what they are, what they do, so on and so forth. Nima will touch on an overview of specialized search interface. We have a all fields interface that is available to you, and then we'll talk about some additional features. Then later, after the break, you'll get an overview of the Immunome Browser. We've introduced that to you. Bjorn has showed that to you, and Randy has also talked about it, but you'll get an introduction to it, but then you'll get a more thorough overview of it by Julia. And then after that, we'll go through some static screenshot examples of queries, so you get an idea of the steps that you would take to do some queries. And then finally, in the afternoon, we'll do this live. So we'll do some exercises that we'll take you through, and then also we'll take ones from you, so we can do those live as we go. So detailed overview. So this will be new to some, familiar to others. So the first thing I want to mention, as Bjorn, or go through again, as Bjorn mentioned, is we're in transition. So starting in December, we are going to launch 3.0 IEDB search interface, which is visually and functionally distinct from the current one. And at this time, what I want to do is get an idea from you folks. How many of you have used the current site? Show of hands. Familiar with the current site? How many of you have looked at the new 3.0 site? Okay, good, good, good. So it's a little bit of each. And how many of you have nothing? Newbies, shall we say? Okay, good. Well, as Bjorn mentioned, this is the current site here. If you click right here, that'll get you to the beta site, and you can play with that. And for those of you who have not already done so, you can check that out. But that's what we're going to be talking about for the whole part of today. So this is what it looks like, and Bjorn again has shown this to you. Again, this is functionally and visually very distinct from the current site, the 2.0 site. And what I've highlighted here in yellow, red, and purple essentially are the three main sections. This is about the IEDB. These are the basic metrics that you can see at a glance, how much data is in there, how many papers, how many epitopes, non-peptidic, peptidic. The center feature here are the search panes, or the search boxes that we have that are there, and I'm going to go through those in great detail. And then over here, you see upfront access to the tools, which is really important. So a lot of people come to the database nearly for the tools. So that's up here. This is going to be for tomorrow's talk, so I'm just showing you where they are on the search interface, or on the home page, sorry, but they will be discussed in great detail tomorrow. Then at the bottom here, and this will be discussed by Debbie later, you can get feedback, there's help requests, there's a solution center down here. Down here you see a date for how frequently the database is updated. And then up here you see the usual, the menus that are similar to the old site, which is you can get at specialized searches, you can go to the analysis resource, keyword search, and again, you'll learn more about the help from Debbie later on. So the main feature of today is going to be discussing the search interface. So the main section of the new home page is these 6 search panes or boxes or sections, whatever you want to call them. What you basically see is that you can search by epitope, you can search by antigen, by host, by disease, by restriction, MHC restriction, and also by assay. This not only reflects, as you saw nicely in Randy's talk, how the data is organized, how we conceptualize it, and how it's captured based on what comes out of the papers, but it also hopefully reflects the things that users are most interested in getting at. Whether it's at the disease level, the level of host, the level of peptide, you can get at it using this search interface. Now, it's important for me to point out here that these search interfaces are available not just at the home page, but also within on the results page, because they're present in more than one spot. On the results page, as you saw earlier with Bjorn's in Bjorn's talk along the left-hand margin of the results page. And this is just a snapshot. I've cut it off. What you see along the left-hand margin are the same panes. This allows you to refine the search that you've done, but it also has some key elements, and I'll be discussing those later, that allow you to do some more specific, more complex searches as you go. So the next series of slides, I'm going to walk you through each one of those search panes and just highlight the main features of each of those as we go. And of course, I've taken a snapshot of each of the 6, and that's why you're seeing them in isolation. But normally you'll see them obviously in an aggregate on the home page. So what you see here is a reflection of one of the most common queries that people perform in the database is a peptide search. So as such, you are able, in this box, you're able to search by peptide sequence. You can type that in. You can also search by the type of peptide, the conformation of the peptide, whether it's continuous, discontinuous, linear, by the chemical type. You can look for peptidic ones and non-peptidic ones, and you can also search by homology. So this in and of itself is a very powerful set of tools available right up front on the home page. The antigen. Now, we've talked a lot about antigen, and I want to take a few seconds to explain what we mean by antigen. You hear that word all the time, but in the database, we use it to describe many things. An epitope can be an antigen, right, if it's on a plate. A protein can be an antigen, right, if it's on a plate, but also an organism. So the antigen field allows you to get at different types of antigens. An organism antigen that is an organism antigen that's also a protein or a molecule. So you can type in a pathogen, it's a virus or bacteria. You can type in, if you're interested in autoimmune, it would be a mammal, a mouse or a human, self-antigen, but also the source of an allergen, graft or something like that. That can be typed in at this level, or you can type in here. And all of these are text boxes that if you click on, you can start typing. And if you do that, we have an autocomplete function, as you see in this inset down here on the right. For example, if you start typing HCV, it'll be a list of possible matches for that, what you're typing in, and you can click on that, and that'll save. That's a big time saver, and also gives you an idea of what's in there, which is kind of nice. Now, as is alluded to in the previous 2 talks, while we are an epitope database, we are very much also a database of assays, because we capture every context in which the epitope was defined. So a paper could be all about B cell, a paper could be all about T cell, a paper could be both B and T. So we capture all of that information because some epitopes are B and T epitopes. So you get the idea. This finder allows you, this interface allows you to search at the high level, all T cell assays, all B cell assays, all MHC ligand, which would be binding or elation. The default is all of them, and also for positive, but you can also look for negative data, and you can mix and match. You can look for just B and T with no MHC, you can look for just MHC, so on and so forth. You can also type one of interest into these boxes, and if that doesn't get it done, you can click preview, and I'll be going through that later to show you examples of how that works. Now, the interesting thing about T cells, as many of you know, is T cell responses come in different flavors. You have class 1, you have class 2, you have non-classical. This search interface allows you to get at it at this level, so you can use the radio buttons, again, like many of the radio buttons you've seen in the past. Select class 1, class 2. You can also type in an allele, specific allele of interest here, or if you can't find it, use the tree view, and again, I'll be going through those. The host is obviously a human or an animal model. That's the host. So it's something that's immunized, something that was sick, something that was administered something. That's what the immunological context is that's being defined. It's not an epitope unless it has a host, right? So on the home page, you can use these radio buttons to search at the high level, so humans, rodents, non-human primates. There's also this drop-down menu with a convenient list of common ones. But if you have something very specific in mind, over here, you can open up the Finder and use that to find something very specific, a very specific mouse strain, a very specific rabbit strain, like transgenic mouse or something like that. That can be, all of the data for those particular things can be accessed in this way. And search by disease. This is a very unique aspect of the database. This allows you to look specifically for data per disease, but it is, that's very important to point out, the clinical status of the host. It's not a categorization that we've done. It's actually clinical. So if there's a patient history, or the paper has defined that it's an animal model of EAE, for example, like an autoimmune situation, we can capture that and you can search by that. So you can hunt for that. It's really unique because you can look for things like autoimmune and allergy, which are kind of distinct. You can look up influenza, and that's influenza. That's the disease, right? But for autoimmune and allergy, it's a little bit more tricky. But so you can look up at those higher levels for allergy epitopes and autoimmune epitopes. It's really neat because you can also do things like you compare data between the human and the animal model of it, which is kind of cool. So this completes walking you through the 6 panes, so all the 6 search panes, shall we say, or search boxes that are on the home page. Now I'm going to transition to showing you, you've already seen this with Bjorn, which is a very good introduction into this. This is the results page. So if you run a query, this will be the next page that you see. Let me just walk you through this. So again, you see the search interfaces repeated over here on this side along the left-hand margin. You see the data in a table form, and each table is delineated by a tab. So you see epitopes, antigens, assays, and references. So if you click on each one of those tabs, you can see the data in that. Also present is this quick glance summary in parentheses of the total number, or the query you performed, the total number of epitopes, antigens, assays, and references. And then the other really neat thing about this, and I'll show you, we'll get some more features covered, but over here you see 2 columns, one number of references and number of assays. The thing that's nice about this is you can see for a particular epitope, the number of references and the number of assays captured for that. So it gives you kind of a relative abundance at that level for a single epitope. So you can look at that, whether it's well studied, whether maybe that's an immunodominant one for a particular autoimmune situation for mice, that sort of thing. It gives you a quick glance at that. And then down here, my goodness, this is hard to use. Down here, you'll see these little Excel icons down here at the bottom and up at the top. For each page that's open, if you want to download that into an Excel spreadsheet, you can by clicking that. And then if you click antigen up here, similarly, you can get the antigen up there. I think it's because I'm holding it. There we go. Sorry, it was how I was holding it. If you click that to open it, then you click that to click the Excel icon to get all the antigen information, similarly with assays and references. So I'm going to transition now from showing you the basic elements and features of the database to explaining some of the functional aspects of the database. In the past several slides, you've seen it, I've either mentioned it or you've seen it, this idea of data tree or tree view or finder. And what I want to do is take a little bit of time to just define those things for you. What do we mean by these things? A finder is a unique feature of the IEDB that provides standardization and hierarchical organization of the data. And what this does is it allows the user to search for an enormous, or find, hence the name, an enormous amount of data at any level of granularity. So all this massive amount of data we have, you can search for just human, IgA, neutralizing, assays. You can do that kind of specificity, and these finders and these data trees allow that to happen. So a data tree essentially is what you would see on your desktop if you open a folder. You open the folder, and then there's things within that folder. That's how it works. It's a hierarchical, ontological, or taxonomic-based organizer with a parent-child node relationship. And I'll show you some examples of that in case that sounds like Swahili to you. But we use this to organize a tremendous amount of data. Here you see it's used for antigens, for all the protein. We have a protein tree using UniProt and SwissProt identifiers. As Randy told you about, we have a non-protein tree organized by KEBI, data and nomenclature, organisms, NCBI taxonomy, allele trees, assay trees, and disease trees. And what's really important for you guys to understand is that while these work behind the scenes to make it all possible, they're also accessible to you. So you can go in and look at these trees and you can see all of the data that's there. And I'll show you this as we go. So how do you get at them? So from the home page, this is again the snapshot of the center of the home page, click search, and here you are on the left-hand side of the results page. If you mouse over these panes, they expand. And as they expand, it reveals access to the tree. what I'm going to do in the next series of slides, starting up at the top here, I'm going to walk you through the finders. And some of it will be redundant, so I apologize for that. But what I'm meaning to do in each instance is highlight what's unique about each finder, and kind of in the iterative process, you get very used to the process of going in, opening it, and what it will look like, and hopefully how to use it. Okay, so the first one you run into is Non-Peptidic Finder, and that's up here in the Epitope search pane. So if you could not find what you wanted and clicked Open Tree because you're searching for some, and again, this was discussed in earlier talks by Randy, by non-peptidic we mean something other than protein, so metal is a good example, a drug, LPS, those sorts of things, carbohydrates. If you click the tree view here, you'll open up this finder. So this is what it will look like when it opens up. And you'll see three basic sections here where you can enter a free text field where you can type. You can also enter an ID if you know that. Down here you see the data tree, which is expandable, and you can look by chemical group in this case. And over here you see current selection. So if you had already performed a search and you had previously inputted a search criteria, it would show up there. Also, when you select one, it'll show up here and you click apply, but we'll be showing you how to go through that live later. So also I want to point out is in the series of slides here, I'm going to be showing you the search box in its expanded form, so you understand why it may look a little different than what you had seen previously. So they're all going to be in there. All right, the Organism Finder. So again, if you come up here and typed it, and try to type in something and you didn't get it, you think, okay, well, I better go to the Finder. So click the tree view to open the Finder, and again, you'll see a very similar format. Here again, you see a free text field to type in, click Find, or down here you can search by organism, bacteria, virus, eukaryote, so on and so forth. The organism, again, just to reiterate, when we mean organism, we mean it can be the epitope source. Organism can be the epitope source. Oh, I'm finally slamming. There you go. Sorry. Epitope source, the immunogen, if it's an organism, but it also can be the assay. So there's these three sections. It has to do with the epitope because it's the epitope. It came from the epitope. The epitope came from it, sorry. It could have been the immunogen used in the assay, but also can be the antigen used in the assay. So that's how you can get at this information. Again, as I mentioned before, it could be an organism or an antigen. So say in this case, I'm going to show you now a data tree. So say here you type in HCV and click Find. What's going to happen is down below, you're going to get a list of options of organisms matching that. In this case, HCV at the top, you see the generic one. So you can either select it by clicking Add, and it'll appear up here. You can hit Apply, or alternatively, you can select Highlight and Tree. And this is going to show you where it exists in the data tree, which is kind of a neat way to see what else is there. So this is the data tree, or in this case, this is the organism tree. So here you see, under the family that it belongs in, then CPI taxonomy again, HCV, the generic high node. Underneath that high node, you'll see all 6 genotypes. If you click open the first one, you'll see the subtypes, and so on and so forth. You can drill down to strains, very specific strains. So this gives you kind of an idea of what the data trees look like. So the molecule finder. So again, within the antigen search interface, you can look for a molecule of interest. Why do we call it molecule? Why do we use that word? Well, that's just to distinguish it from organism. So the protein is the molecule that comes from the organism. That's as deep as it needs to go. But again, the molecule can be the protein that the epitope comes from. It could be that mice were injected with the protein, or it could be the protein was on their ELISA plate. That's what we mean by antigen. So again, you can come down to the search pane. Once you click the tree view to open the finder, you'll see that you can either type in the protein name or an accession if you happen to know that. You can also search for proteins by the organism, which is a unique feature of this. And again, you'll see the tree down below before. So I'll show you examples. So say you're interested in searching for, you're interested in HA, but you want to see what's in there for flu because obviously there might be a lot of flu data. So say I type in influenza and click Find. So again, I get this list of options, proteins that come from influenza down below. And I can either select an individual protein. But remember that, it's important for me to point out that an individual protein in this list is an individual accession. So you'll only get the data for that individual accession. But what might be more interesting to do and more useful, depending on what you're looking, is if you click highlight and tree, you'll get the data tree of all proteins from flu. So generically, all flu, all hemagglutinin. So you see there's this high node that if you click it open, you get all the individual accessions that belong under that node of hemagglutinin. If you click this node, you'll get all hemagglutinin. That might be a nice way to start, broad, and then you go down to more specific things, more specific strains you can be interested in. Because as you'll see in some of the results page, once you do the results, you can see your strains, and then you can filter and select on those ones as you go. So the Assay Finder, if you click on the tree view here and open up the Finder, what you'll see is a couple more options here. And what I mean by that is there's two pull-down menus. So not only can you type in, say if you're interested in neutralization assay, you can type that in, but you also can search by technique or measurement. And let me give you an example of why this is kind of a powerful way to do it. Is you can say, well, I want to look at ELISA data or ELISpot data, or I want to look at a particular kind of CTL assay, but there's different kinds of CTL assays. So, and there's different kind of cytokine assays. If you're interested in just PCR, RT-PCR, you might select just those. If you're interested in just ELISA, you can select just those, or IL-13 or gamma. Again, you can multi-select too, which is very important. And you also can search under the trees. We have these categories of assays, and it's by the type of response, the type of response that's been defined in the assay. That gives you kind of an example of that. The Allele Finder, similarly, is useful because once you click it open, you can search at a couple of different levels. And this is just to find the data. You can say, I'm not sure what's in there. I'm not sure what might be useful. So I can say, I can come down here and say, click human and say, give me all human. Let me see what all the human class 1 data is, and hit find. You'll get a list of all human class 1 alleles that have been captured today, so that you can see them at a glance. And then you can select them and hit apply, and then carry on your search. There's currently 15 different alleles from 15 different species right now, which is actually fantastic. And also, it's important for me to point out here is MHC restriction is often defined in the assay, right? But it's also sometimes, if it's human data, they'll type all the human subjects we have been typed. We can capture that too. So it's used in that respect too in the assay. And again, you can search on that. You can search for the HLA type of the human host that was studied. The host finder doesn't require a lot of explanation. This is what it looks like if you click to open it. I just showed by example here, you can either type in human. If you happen to know the NCBI taxonomy ID, you can type that in. You don't need both. One or the other is fine. But again, you can search by the tree, or you can hunt typing in human and click and see there may be different asks. For example, rabbit would be different kinds of mice. The disease finder is actually very useful in that naming of diseases. What you might name a disease might be slightly different, so you might have a little bit more difficulty getting the autocomplete to work when you type in the text. Contact dermatitis is the example I write here, so that's pretty straightforward. If you click find, you're going to get hits for dermatitis, and you can select from those. But there might be something different. For example, you might be interested in shrimp allergy. When you type in shrimp allergy and that doesn't come up, well, you can use a tree to find that, oh, well, maybe it's under food allergy. You do have a category of this food allergy. That's kind of an example of how you can use the tree for diseases which nomenclature can vary a little bit. Okay, and then finally, at the very bottom on the left-hand side is the ability to search by reference. So very straightforward. Anything here, the title, the PubMed ID, the author name. If you're interested in one of your colleagues' paper or your own paper and find out if that's in there, that can be done too. And this is a kind of a neat feature that we have. Along the very top of the home page is the ability to search, do a keyword search. And it's like a Google search where you put in a term and get back results of that term. And there are the advantages. You can search for a very specific term, like you can search for epitope spreading or mimicry. So that's really neat that you can do that and search certain fields. The disadvantage is because it's searching not all of the fields, you can't be slightly sure that you'll get back all data, but you also get back a lot of it. So that's why, for example, if I was going to do something like epitope spreading, I would absolutely put quotation marks around it, because if I just write epitope, it'll give me back everything that said the word epitope, which would be pretty much all of it, right? So you get the idea. And then I think we're going to turn this over to Nima. Nima will be discussing specialized cases. Okay, it's going.
That audio. Awesome. All right. So good morning, I'm Nima. I wanted to just give you a brief kind of status check of where we are. So as mentioned, I'm going to walk you through the specialized searches in a moment, but we're rapidly approaching the break, so I know many of you are ready for that. What I'm going to do is go through a few slides and then briefly turn it back over to Carrie. And she's going to give you a little bit of a teaser on the Immunome Browser, which is a feature that we're very proud of. Bjorn briefly alluded to it. It's basically, these aggregate response maps that we generate based on the data. That will be presented after the break by Julia. So we give you that little teaser before the break, so you don't go home. Hope to keep you here at least that long. So the specialized searches, as you've seen now a couple of times, from the home page, there is a menu up at the top called Specialized Searches, where basically you get a selection of different topics. And these can all collectively be essentially grouped into 3 clusters. So you'll see the first four are called details: epitope, T-cell, B-cell, and MHC detail searches. The detail searches, or what we currently on 2.0 sometimes refer to as our advanced searches, in quotes, are search interfaces that are designed to give you the capability to search each and every field in the database. So what you've seen till now has been essentially a concise, boiled down sort of a set of fields that one can access by and large most of the data, but not all of it directly. So it's in a way designed to allow you to ask maybe more deeper questions, complex questions, and I'll walk you through some of those in a second. We also have what you see here called the Identifier Search. The Identifier Search is essentially just one way of accessing data using unique IDs, either IDs that we assign to our data internally or IDs that we get from other resources, for example, PubMed. And then lastly, I'll walk you through some of the browse by searches, which are essentially ways of looking at data that's organized in trees, hopefully in a logical manner, very similar to the tree we've seen. But the hope there is to be able to kind of navigate through these trees and drill down to data instead of performing a query and searching for it in that manner. Oops, sorry. Let's do it this way. Okay, so I'll start by kind of, again, reiterating that these specialized searches, the details pages, are intended to give you access to each and every data field. And one of the things that makes the IEDB unique is the fact that our list of data fields is so extensive and deep. And it's, as mentioned before, a result of that assay centricity of our database. We seek to really capture everything pertinent that we think an immunologist might want to know about that assay, whether it's, for example, the isotype of the antibody, the phenotype of the T-cell, and so on. So that is a double-edged sword because, as you can imagine, the number of fields can quickly get out of control in terms of their number. So what you'll see if you go and click epitope details, for example, is a form that looks basically like this. And there are, in some of these detail searches, upwards of 100 fields. So what we've done in order to both not overwhelm the user but also make this somewhat logical is we've clustered these fields into various collapsed clusters. And you can see those fields are separated by these blue banners, which give them a logical description of what they deal with, for example, the reference and epitope that you've seen. But also within those are fields that can be further opened up by clicking the plus sign, and they're kind of accordion collapse. So you have the reference, the epitope fields, and in this case, one example of something that you can access through an epitope details search that you can't access from the home page would be the third one at the bottom called the epitope-related object fields. So as you all are aware, not every sequence that has immunological reactivity is going to necessarily have a natural source, for example. So in those cases, for instance, we could take the epitope, if the epitope is a mimetope or an analog of a naturally occurring structure, we could relate those two together so that it gives you another avenue of accessing data via epitope or analog of natural data. So we can walk through, what I've done here is kind of just try to contrast for you what you'll find here that you won't find on the homepage search. I won't perform any extensive queries, at least not here, but I think the best way I would advise you to kind of familiarize yourself with it is to just basically go on a scavenger hunt and look at it and see what's there. And then in the breakout sessions, we're happy to sit down with you and help you use it further if you'd like. So the second type that we have is called the specialized search for the B-cell details. So these are all data fields relating to B-cell responses, B-cell epitopes. Again, you see common themes with the reference and epitope panels up at the tops here. But the two red circled sections are new and kind of unique in this case to the B-cell assay details. Bjorn had mentioned at the top that we strive to capture, for example, how the host was primed, how the reactivity came about in the first place. So in that first section called immunization, which is circled red, you can see a field there called in vivo process. And what we've done here is basically laid out those process types that, again, Bjorn had mentioned, that are essentially choices for how the priming was done, whether it was an administration in vivo, a physical immunization, for example, whether this host had an occurrence of an infectious disease. So this, again, is a multi-select field like others that Carrie had shown you that allows you to be very pointed in your question formulation. If you're interested solely in infected donors who mounted a response, who had some infectious disease, you could delimit the data by saying, I'm only interested in responses where the host had an occurrence of infectious disease. Furthermore, in the B-cell assay section below that's also circled in red, we provide a number of fields, again, mostly collapsed under these plus signs, but they can be opened and revealed to, again, show more database fields and really exploit the power of the IEDB further. So on the next slide, I'll show you one example. What the next slide is going to do is basically zoom in on that B-cell assay section. And what I've done here is I've opened the section entitled Assay Antibody to reveal a host of more fields that were kind of collapsed under there. Excuse me. And in this case, I've put this circle, red circle, around a couple of fields that are available here but not available elsewhere on the other search interfaces. So the question, for example, that came up earlier, I believe Professor addressed in you asked if you could search by isotypes. This is where one would specify the isotype or isotypes of interest, and that would immediately delimit the results down to only those cases where the paper or the data specifies that this was the isotype that reacted. And again, it's a multi-select list. You can see it's really extensive. We go down to isotype of every nature. And then further, just above that is a field, a free text field called the assay antibody name. And many of you may be interested in searching for data for a particular monoclonal antibody, for instance, that has a very common name for instance, that's given by the manufacturer or by the lab that produced it. And while free text searches are always hit or miss, that capability is here so that you could put in a name of a common monoclonal and see what you get. But again, just an example of another field that's not available on the homepage search. So moving to the T-cell detail searches, it's almost a mirror image of what you see on the B-cell details pages. But of course, the fields are going to vary somewhat. So again, you have this kind of similar structure where there's an immunization section and a T-cell assay section. And to kind of give you an analogy to what I did on the B-cell side, if you focus in on the T-cell assay details section, you can also define, for example, the phenotype or the type of T-cell that you're interested in. So this is available through drop-down lists contained within that effector cell cluster. So in the effector cell cluster, we capture several different parameters. One being, first of all, what were the effector cells or T cells, whatever they may be, what tissue were they isolated from in the host? So you can, for instance, specify that using this first drop-down, and in this case I've chosen blood. If you are solely interested in CD8 T cell reactivity, you can specify that in the next field. Again, these drop-down lists have dozens of selections, so I would just encourage you to look at them. I'm sorry that all the choices aren't displayed here, but we have CD4 and so on. And then lastly, there's a field called effector cell culture conditions, which is open in this case, and you can see it has a series of selections which essentially seek to use immunologists, lingo, which specifies essentially how were these T cells treated before they were assayed. So some scientists are interested in seeing reactivity of T cells that were taken directly from the host and in no way re-stimulated, for example, before they were assayed. In that case, you could use the first selection direct ex vivo. Sometimes they are re-stimulated and you're looking at the reactivity of a clone or a cell line. Those can be selected as desired as well. Again, multi-select list there. So moving along, what I did was I wanted to kind of go back to the homepage search and the theme that we introduced you with which was that of trying to make the homepage search, reworking it in 3.0 to make it a little bit more user-friendly, to enable it to be inclusive of what we think are, the vast majority of user queries. But also, we have future plans to transition what we call the detail searches toward the same look and feel, although they currently aren't. You may have noticed that those, or if you use the site, you may notice that if you do a specialized detail search from 3.0, essentially what it does is it takes you back to the advanced searches of 2.0. And that's because they haven't yet been revamped to match the look and feel of 3.0. But what I have here is essentially a snapshot of the direction we're moving in with the detail searches. And not much will change other than the layout, in that you will still have access in a detail search to every single field in the database, only they will be buried in these side panels that Carrie has shown you, instead of being buried in accordion collapse fields on a top-down search page or a website page. Again, you can perform your search here by specifying effector cell phenotype, B-cell, antibody, isotype, and hit search, and these results, meaning the number of epitopes, the antigens from which they come, etc., will get updated and refined as you go. So we see these really as like refinement panels as much as they are search panels. Okay, so that's a future. I don't know if anyone knows the timeline, but for now, in the foreseeable future, all detailed searches will be done on the previous interfaces that I showed you. All right, and so the second kind of specialized search I had mentioned was the identifier search. Very briefly, I'll just show you two. if you click on that link, you'll get this set of search boxes. And they essentially can be broken up into two clusters, those that are called IEDB identifiers and those that are called external identifiers. As I mentioned, we assign identifiers, but we also link to other resources that have their own. So if you're interested in a particular, for example, reference, PubMed ID, and you know that you have this manuscript in front of you, you see that it has epitope data, for example, but you want to see it in a concise, reported way, you can put that PubMed ID in and quickly link to the summary of that epitope-related data that we've curated. Do this with your own publications as well. Again, as Bjorn mentioned, we think that we've got essentially 98, 99% of published epitope-related data in our scope out there. But if you come across anything that's not and you feel it should, we're happy to hear about it. Secondly, I wanted to just show you the one beneath, which is the PDB ID search. As it has been mentioned a few times now, we do have a 3D structure kind of specialist crew in here. Julia and Debbie specialize in capturing that. And so whenever a immune receptor has been shown to interact with an antigen and the minimal data requirements are there, we capture that, and then one can view that interaction in what's called our epitope. So if you had a PDB ID in mind and you wanted to see whether we have curated that and hence look at the structure of the interaction between the epitope and the receptor, you can put that PDB ID in there and then drill down and eventually look at it in the epitope viewer. And this basically shows you in color-coded forms what's the antigen, what's the epitope, what are the contact residues, etc. And it's rotatable in 3D space, so it's a very, very nice feature. Lastly, I'll just walk you through the 3 browse-by searches. We have an MHC Allele search, a source organism search, and a 3D structure search, speaking of 3D structures. So this is, again, an attempt to collapse the data down into hierarchical trees that can be kind of navigated by opening these plus signs and kind of going down the tree in a logical way. So the first one that I'll show is the Browse by MHC Allele. So if you click on the MHC Allele, Browse by MHC Allele link, you'll see this tree immediately. And it'll be, by default, collapsed down. But of course, these plus signs have lots of wonderful MHC-related data buried within them. And you can see to the far right of each the number of structures that are represented there to have some kind of relationship to that particular organism and then allele as you drill down. So, for example, if we just follow where the red arrows are, I'll continuously open those plus signs and show you that if we're interested in looking at what's available for MHC-related data under humans, we can open that node. And in this case, I've opened the class 1. And under class 1, I've opened A to reveal a host of alleles that each have some data captured in the IEDB for them. In particular, if you look at HLA 0201, I've boxed the number 16,127, which shows you that there are that many structures that are some way reporting data for that allele, whether it be a T-cell restriction, a ligand dilution, or a binding assay, or something of that nature. The nice thing about the numbers is that as one navigates the tree, you get an overview of how much data is available for each node, for each branch. But furthermore, they can be clicked to immediately take you to that data. So if I were to click that 16,000 link, I would basically arrive at a summary page for that allele, which at the top gives you pertinent information about the allele, including a motif map. And then below in these tabs are summarized all of the assays that, again, mention that or tied to that allele in some way, either because they had epitopes that bound or they were the restriction of the T-cell assay, etc. So you can navigate through these tabs, and of course this is a very broad example, so you would have a lot to sift through in that case. Source organism is a similar organization. In this case, however, the tree is a representation of organisms that have been the source of epitopes that are curated. And Bjorn mentioned at the top that NCBI taxonomy is wonderful, but it's kind of too wonderful, right? Because it's got 1,000,001 nodes in it. So what we've done is we've presented a tree that is kind of customized. Its foundations are in NCBI taxonomy, but it's been pruned down to hopefully make it a little bit more user-friendly. The same theme pervades with the numbers of structures being links. However, even though we've pruned it down, it can still be a little cumbersome. And unless you really know taxonomy, some of these nodes might be hard to drill down or even know where to start to go down to the data you're interested in. So we put in a search, pretext search bar at the top. And so, for example, if I wanted to drill down and see what epitopes are found for various flu strains, for example, I could try to do that by opening up several boxes and going very deep into the tree, or I could just type flu or influenza. One nice thing about these pretext searches is that we've made an effort to make them responsive to synonyms in everyday kind of terms. So as Carrie had shown you, you get a kind of a summary table for the word flu when you hit search. And here you can, again, choose one of interest and highlight it in the tree. In this case, I've done influenza A virus. You can see there are 6,000 some odd results there. Pardon, yeah, question, please.
How do you find that the particular strain is multi-drug resistant?
Oh, if it's a multi-drug resistant strain is the question. So the question was, if you put in the name, how would you know whether the results show a strain which is multi-drug resistant? Okay, so I'll give you my take on that, and then I'll also let my colleagues weigh in. In this particular case, unless the name has multi-drug resistant or something that implies it's multi-drug resistant in the name or in the synonyms, you won't be able to know that unless, for example, if it's, what's the name of the TB one? Or MRSA or something that implies that. If the name contains that, then you can easily come about it. But otherwise, any input from you guys, I think you would have to know, from some secondary resource that it's multi-drug resistant, and then try to search for it taxonomically that way.
With that, you can then download that information spreadsheet. That would be a good way. And annotate. There are information comment fields, assay comment fields. You can answer a lot of innovation that way. That's something you could comment on. There's two ways that you can get at it. Other than just another, yeah, it may not be known. It's very, after doing this for about 10 years now, I would say other than this, yeah, it's something you can't get at. Yeah. Yes, that's a great question. That would be something that search can make. We can actually identify people. It is wonderful comment. It is to show you that. They go into to try and show you how often.
Or your protein sequence. Okay, so I'll just leave you all with the final kind of snapshot. And in this case, for the browse by 3D structure, I haven't really opened anything, but I just want you to be aware that it's there. It's, again, a navigatable tree in this case. It's important to point out that these organisms are the organization is by organism that serves as the source of the antibody, T-cell receptor or MHC receptor. So if you're interested in seeking out structures that way, this tree is actually much more abbreviated because 3D data is more sparse. So this is one that you could conceivably just kind of click through and peruse and get something out of without really doing a true query per se. All right, so I think at this point, we're going to do the teaser as promised for the Immunome Browser. This is fairly brief, but then we will, oh, we do take a break. Okay, never mind.